We're doing this because it snowed again. My luck continues. All right, so we're going to pick up pretty much where we left off last week. This is the exercise we were doing, exercise 2, where we were doing a sample annotation from a species, Drosophila erecta. So I'm just going to go down the document here until to page 23, which is a little bit before where we left off. We did start doing what we did on page 23. So, I went a little too far there. All right, so let's see, top of page 23. This is where we were starting to put together our gene model. So we're constructing a gene model by comparing Drosophila melanogaster coding exons of the isoform B of the CI gene to our unknown sequence. So we're going to go to Gene Record Finder, and we're going to pull up the CI gene. So you'll probably want to have your notebook page open. You'll want to have your genome browser page open, and I'm going to go to Drosophila erecta. We're working on Contake 38. You want Gene Record Finder. I'm going to put in CI gene. And you'll also want to have your BLAST page open. So this is what we're looking at on page 23 of the annotation exercise. So we're interested in annotating this gene here. So we're going to start by annotating the first exon. So remember, the goal of this is to use Drosophila melanogaster as a guide to identify the orthologous regions in our unknown genome sequence. So we're interested in using the coding region of this gene. So we're going to start by taking the amino acid sequence of the first exon of this B isoform and looking at the nucleotide sequence of our unknown species and determining, is there a match? So we're going to click right here on our exon. Remember, these numbers here, these are unique exon numbers. So every exon of every gene in Drosophila melanogaster has one of these. So we're going to click on that, and it's going to pull up the amino acid sequence. And we want to compare our unknown nucleotide sequence to this amino acid sequence. So this is where we're going to do a BLAST X search. So we're going to copy this. I'm going to copy it. Oops. Copy. Then I'm going to go to BLAST. I'm going to choose BLAST X. And remember, with a BLAST X, we are choosing to search a protein database. And remember, when we're doing these BLAST searches for our annotation, we are going to compare two known sequences. We're not comparing our sequence to a database, but two known sequences. So we're going to click on Align two or more sequences. And we're going to paste our amino acid sequence in the second box. Make sure you include this top header. And in here, we're going to choose the unknown sequence that we downloaded from eClass. So if you don't know where that is on your computer, you can go to Annotation Resources, and I am actually going to upload it right now. Apologize, I thought it was on there. So I'm going to upload the sequence. This is going to be the erecta Contake 38. And now it is right there. Let me just set it to download. So now if you click on this in the eClass, it'll just download this text file. So if we go back to here, you're going to take that text file and you're going to upload it to your BLAST. So it's the same file I just downloaded. Now, actually, I'm going to change this to a rich text, a different format. You won't have to worry about doing this because you will be provided these files. So you're going to use that. Again, direct to config 38. Sequences in here. We're going to go to algorithm parameters, and this is on page 26 of your annotation exercise. And just a reminder, if you didn't bring that home with you during the snowstorm, it's right here on eClass practice annotation, so you can always download that. So going back to our BLAST here, the two things that we're going to want to change is we're going to go to compositional adjustments and we're going to turn that to no adjustment. This is not in the exercise handout because this was a newer change on BLAST on the website. And then we're going to unclick low complexity regions. And we're going to click BLAST. And it, for some reason, didn't like what I did here. So let's try this again. All right, I'm going to go offline for just a sec to figure out. I think I fixed the problem, so I'm going to pick the file. This is the same one that you would download. Sorry, you would download right off of the eClass here. I'm going to pick it. I've already set my changes here. So we're going to do a BLASTX of the Drosophila erecta config 38 against this exon here. And we're going to click BLAST, and it should go pretty quickly. So again, it always shows the relevant information up top here. So we took 53,840 nucleotides, translated it 6 ways, compared it to 17 amino acids. On the picture here, the graphic summary, you're going to see just a really little dot because we're only comparing it against 17 amino acids. So where it matches is going to be pretty small. And our alignments are going to be down here. And you can see that there are only 2 matches. And we wouldn't expect many matches here because we're looking for a specific exon. The top one has a pretty good E-score, 3 × 10⁻⁷. You can see that it matches up quite well. All 17 amino acids are accounted for. It's in frame +3. So, and just also keep in mind, this is all in the annotation exercise handout, so you can read along as we're going through this. So what this is telling us is that we should be able to find this exon somewhere around the coordinates of 15,405 to 15,455 in our unknown sequence. Since this is the first exon, we should expect to see a start codon around here, and at the end of the first exon, we should expect to see a 5' splice site around here. So we use BLAST to hone in on an area where we think we're going to find the coordinates, but we still have to use our genome browser to get the very specific information. So now on page 28, we're going to go back to our browser, and we're going to, and first what we should do actually is take a screenshot. I'm going to take a screenshot of this here, and I'm going to make sure I include this because this is showing me what exon I am blasting. So I'm going to take a screenshot here, just like that. I'm going to go to my notebook, and under notes, I'm going to paste it in, and I actually have I already pasted it in previously. You can see it right here. So the way I did that was go to Insert, Image, and then pick, and wherever your screenshots go to on Macs, they go to the desktop. Just go ahead and insert that in. You'll notice that I've given more information. So this section is the CI gene analysis, and these are the blast results for isoform B, exon 2, underscore 868, and so on. So we're now going to go to approximately this region in our browser to see if we can find the start of this exon. So we're going to go to the browser here. We're looking for 15405. So that's going to be somewhere around here. So I'm going to zoom in by just making a box with my mouse, like so. And I'm just going to zoom and make sure your base position is set to full right down here. Refresh. I want to be able to see my bases. I'm just going to zoom in a little more because I want to be able to see the individual amino acids. And I'm going to zoom in just a little more so that I could get 15405 in there. And there we go. So I'm going to zoom in just a little more just so I can walk you guys through how this works. So up here you can see the nucleotides. So this A is 15401, this T is 15402, this A is 15403, and so on. So the way the numbers work is you have this line, the number and the letter are to the left of the line. So this represents this nucleotide, this is 15403, 15404, 15405. So we know around 15405 we should see a start codon. And what do you know? A, T, G, and that makes a methionine. So we see a methionine here in the third reading frame. And we should, according to our BLAST, find our match in the third reading frame on the strand. And in fact, +3. Great, that makes sense. So it looks like this A right here is likely going to be the first nucleotide of the first coding exon. You'll see that there's some other evidence that is useful here sometimes. For the start site, you can see that there's RNA sequencing data. So these different colors represent RNA sequencing data. So they took all the transcripts from this particular species and they sequenced them. We'll talk about how later in the semester how this is done. When you see any sort of peaks here, and I'm going to zoom out so you can see the peaks a little better. Whenever you see peaks, that's indicative of it being a gene because that region is being transcribed. Now you'll notice that here there are still peaks to the left of this ATG. And the reason for that is that most Drosophila genes have a 5' untranslated region upstream of the start codon. We're not going to be annotating that. We're interested right now in just annotating the coding regions. We'll worry about the untranslated regions later on. So what we've got here is we've got our first nucleotide at 15405. And this is all explained on page 28 as well. So we're going to go to our lab notebook. We are going to go to our CDS worksheet, and that means we also have to open up our Google Sheets. and we need to go to our CI workbook. And I'm actually going to clear this out because this is from the other day. Close your eyes, you don't want to see the answers. So we're looking at this exon here, 28680. The ortholog start site is going to be 15405. And that's going to import into our lab notebook. There we go. So the translational start site is generally pretty easy because you're looking for a methionine in a specific area. Next, we want to find the end of this exon. So we go back to our BLAST, we should find it somewhere around 15455. So we're going to go back to our browser and we're going to zoom out a little bit. So I'm going to zoom out 10x. And then you can see 15455 is somewhere right around here. So I'm going to zoom back in, and it's going to take a little while for you guys to get the hang of the zoom. That's fine. You're going to have to play around with it. Now, actually, even before we do that, I'm going to zoom back out. We were looking, the start site was right here. So we just annotated our start site here. This is approximately where we see, we expect to find our 5' splice site. We know that this exon is in frame 3, so one thing that we oftentimes want to check is we should make sure there are no stop codons anywhere in frame 3 between the start site and the 5' splice site. And this will be the same thing for subsequent exons when you make sure there is no stop codon between the 3' splice site and the 5' splice site. So let's zoom in this 15455 region. I'm going to zoom in a little more. So when it comes to splice sites, remember 5' splice sites 90 to 95% of the time are going to be GTs. So right away we're going to be looking for a GT in this region. And we've got one right here. Cool. So is this our actual splice site? Well, it's pretty close to 15455. It's not exactly there because we know sometimes BLAST is incorrect. If we look at, if we consider this as our potential splice site, the gene prediction programs, so if this is our potential splice site here, it means that, remember, the splice sites are in the intron. So it means this G would be the end of our exon. So if that's the case, then the gene prediction programs here, we've got our gene prediction programs open, GenScan, TwinScan, SGP, all predict that the exon ends at that G. We can go down to predicted splice sites and set that to dense. And you can see that here. I'm just going to click on that. It's going to tell us what that means here. This is what's called a medium confidence splice donor. So a splice donor is the same thing as a 5' splice site, and a splice acceptor is the same thing as a 3' splice site. This means that there's a pretty good confidence that this would be an actual splice site. And this is just based upon sequence. This has nothing to do with any sort of data from genes being transcribed or anything. This is simply based upon the sequence of the genes, of the genome. So we've got that as evidence. We've got down here RNA-seq data. So remember, and I'm going to zoom out a little bit. So remember, RNA sequencing data is going to correlate with coding regions because they only sequence the transcripts that have been processed, so the introns have been removed. So if you see RNA sequencing data where it drops off right here, there's a pretty good chance that's the end of an exon. And then we also have this top hat junction track here. This is where they're predicting splice sites based upon the sequencing data. So unlike these predictions, which are based solely on the sequence, this prediction is based upon the RNA sequencing data. And this prediction also says this would be the end of the exon right here. So I'm going to zoom in a little bit. Sorry, I'm pressing zoom out. So all of these are saying this is the end of the exon. So this would be 15, 4, 5, 6. We've got a pretty good amount of data that supports this. So we've got our gene prediction programs, we've got our splice sites, we've got our sequencing data, we've got our top hat. So all of this is pretty good data to support this 15, 4, 5, 6 as being the end of our exon. So I'm going to go to our Google Sheets, and for right now I'm going to put 15, 4, 5, 6 in there. Now, remember, this wasn't the same as our BLAST results, so this is why we have to annotate by hand. BLAST is never going to get it right, or BLAST is never going to always get it right. So it's going to ask what strand this was on. This is on the positive strand, +3. So plus, and it's in frame 3. If you're trying to put in a plus, you might have to put in an apostrophe because it wants to put in a formula. And then frame 3. And the last thing that we put in here is the phase. The phase has to do with where that splice site is in respect to the codons. So they talk about this on page 30 and 31. Highly recommend that you read this because they do a good job in kind of explaining this. And they also talk about the RNA sequencing data on page 32. So also highly recommend reading that as well. So since we're not in the lab this week, definitely want to read these things. So what does the phase mean? So let's go back here. We know that this is in frame 3. So if this is the last nucleotide here, we can kind of draw a line down like this. And you can see in frame 3, so imagine there's a line right here. In frame 3 you have a codon that codes for this amino acid, phenylalanine. You have a codon that codes for this amino acid, histidine. And then there's one nucleotide into the next codon, and then it's the end of the exon. So there's one nucleotide just kind of hanging out. It's not a full codon. This is what we're talking about when we talk about the phase. This is what we call a phase of 1, because there's one additional nucleotide. And this is explained here on page 31. I'm going to jump down there. And they start talking about this on page 30. But I'm going to refer to the picture. So it is common, very common, for exons to terminate in the middle of a codon. So this is an example very similar to what we're looking at here. This is an example of the exon ends in the middle of the codon, and there's one nucleotide overhanging. So this is in, this is in frame 3. We're going to look at the next exon next, and we're going to see that that is in maybe frame 3, maybe frame 1, maybe frame 2. But we need to take into account what the phase is, because the next one, the next exon, wherever that 3' splice site is, has to be so that the next exon is in phase 2, which means that there are 2 bases overhanging before the first full codon. When this happens, this 1 and these 2 are going to form a codon, and it keeps this protein in the correct reading frame. So here you see a phase of 1 will match up with a phase of 2. Now, if you can imagine, if there were 2 nucleotides overhanging here, you would want to match that up with 1 overhanging here. Or sometimes the exon ends at the end of a codon, which would be a phase of 0, so that next exon needs to start at the beginning of a codon, which is a phase of 0. So this is kind of summed up in this chart here. If the donor phase is 1, which is what we have, then the acceptor phase, the next exon, has to be 2. If it's 2, so if there's 2 nucleotides overhanging here, there needs to be 1 in the next exon. And if the donor phase is 0, then it has to be 0. So basically, these have to add up to either 3 or 0. And that's how they stay in frame. So if we go back to our gene here, if this is the end of our exon, this is a phase of 1 in here. I'm going to put phase 1. That way, when I start annotating the next exon, I'm going to know that the next one has to be in a phase of 2. So this is kind of a placeholder telling me this next exon better start with a phase of 2. So we've gone ahead and we've annotated the first exon of this gene. We've annotated from here to here. So what's the next step? If you look at page 31, there's two 30s and two 31s because Microsoft Word and I don't get along very well. The next step is we need to now annotate exon 2. So how do we do that? We go back to gene model or gene record finder. We're going to get the amino acid sequence for exon 2 of our B isoform. And remember, this exon 2 is going to be the same as this exon 3 and this exon 3. So we're going to click on that. We're going to copy that amino acid sequence. We go to BLAST. If we want to just rerun this with a different amino acid sequence, we can click on Edit and Resubmit. And we simply clear out this amino acid sequence and we put in this amino acid sequence. It'll keep all of our parameters, and then we're going to click on BLAST. BLAST results here. Again, same query length because we're using the same unknown nucleotide sequence from our species. And we've compared it to 213 amino acids from Drosophila melanogaster. And again, a short match, as we'd expect. And here there are 1, 2, 3, 4, 5 places that this amino acid sequence aligned. But you can see most of them were not very good. So this second one was only a handful of amino acids, pretty bad E-score, and they get even worse from there. So everything down here, these are just random matches. These are not real matches. Up here we have an E-score of 1 times 10 to the negative 1/15. That's pretty good. It accounts for all 213 amino acids. So we can be pretty sure that this is going to be our exon 2. And interestingly, you can see there are some differences. So our exon, or our sequence, does not have these 3 amino acids. While our sequence has these 3 amino acids, the Drosophila melanogaster sequence doesn't. So this is likely due to an insertion or deletion that has occurred. So you're probably thinking, and actually up here it's 4 amino acids, 1, 2, 3, and then 4. You're probably thinking, well, which was it? An insertion or a deletion? We can't tell. But what we can do, and this is something we might want to do later on after we annotate the entire gene, is we may want to compare the amino acid sequence of different species of Drosophila. And this is what's called doing a clustal alignment. And if we do this, we can actually determine when this change occurred evolutionarily. So at which common ancestor was there a deletion or an insertion that occurred? But we'll talk about that when we get a little later on. Here, we can see this is our alignment. So this would likely be about the beginning of exon 2, and this would be about the end of exon 2. So we're going to look in the area of 15,521. So we're going to go back to our genome browser. I'm going to zoom out. And I don't like what it's doing with these here, so I'm going to change. For some reason, it changed my predicted splice site, so I'm going to move that back to dense. And we want to go to around 15,521. So this is 15,000, 15,500, 16,000. Or I'm sorry, we wanted to go to 15,521. We also probably want to take a screenshot of this. So this is our alignment for our second exon. So we would take a screenshot, go to our, this is the practice annotation, go to notes, and so after this first screenshot, this I've already put in the second one, but what I forgot to type in here was this is BLAST results for isoform B exon 4_868_2. And then you can go ahead and insert this image right in there. So we're going to look for the beginning of this exon. So we're going to go to around 15,521. So I'm just going to zoom in a little closer. So 15,521 is going to be somewhere in this region here. All right, we're getting closer. So this is 15,520. So I'm just going to zoom in like this. So 15,521 is here. Now, we need to look for a 3' splice site. Zoom in so you can see it a little better. A 3' splice site is going to be an AG. It's always an AG. So I'm going to just kind of search along here, see if there's any AGs. Okay, there's one right here. Cool. And I don't see too much in that general vicinity, so right away I can say, yeah, this is this is a potential 3' splice site, which means this would be the potential start of our next exon. So let's look at the data that might support this. Well, one, gene predictions, so GenScan and SGP both suggest that this exon starts right here. For our predicted splice sites. Sorry, let me zoom in so you can see this a little better. So for our predicted splice sites, there's a high confidence 3' splice site right here. And if we look at our RNA sequencing data, the RNA sequencing data picks back up right here, which just so happens to correspond with this A. And the same thing with our predicted splice sites according to TopHat, which uses the RNA sequencing data. It suggests that this is the beginning of the next exon as well. So far we have a lot of data that supports that this A is the beginning of our next exon. So if this is the case, well, first what we need to look at is what frame is our next exon in. And in our BLAST alignment, it showed a frame of +2. So if we go back to here, this is frame 2 is the second row of our amino acid letters here, right here. So if this is the beginning, this would be our beginning of our second exon in frame 2 here. If we draw an imaginary line here, this gives us a 2 nucleotide overhang before the next amino acid. So that would be a phase of 2. If we look back at what the phase of our previous exon was, go to the worksheet, the phase of the previous exon was 1. Now we know that if the previous exon is in a phase of 1, the next exon has to be in a phase of 2. And in fact, that's the case. So all of our evidence here is pretty strong for this being the correct splice site. And in fact, 15519 being the beginning of the next exon. So we're going to call this 15519. We're going to put that in our Google Sheets. Let's go and see if we can find the end of this exon. So we're going to go back here, go back to our BLAST. It should be somewhere around 16156. So I'm going to zoom out and go to about 16156. There you go, 16156. So that's going to be somewhere around here. And there we go. And what data do we have? Or what do we see here? Well, 16156 is down here, so I'm actually going to zoom out just a little bit. 16156 is down here. We're looking for a GT, which would be our 5' splice site. Got a GT right here. That means this would be the last exon of, or the last nucleotide of our exon. It would, the prediction programs say it stops right here. That looks good. The splice predictor programs like this as a splice site. Cool. Fortunately, there doesn't appear to be any sequencing data here. So if we zoom out, we'll see some of these previous exons have sequencing data for part of the exons, but not for all of them. So this was our previous exons. So you have to keep in mind, so there's no sequencing data for this part of the exon here. When it comes to RNA sequencing data, lack of sequencing data is not evidence to not use a splice site. So I guess a better way of saying that is sequencing data is, and I'm just zooming back into this 156 area here. So sequencing data is a good confirmation. Sorry, I'm just trying to get the right zoom. Sequencing data is a good confirmation for a splice site, but lack of sequencing data doesn't tell us anything because some areas of the transcripts just don't get sequenced very well. So all of that considered, this GT looks like a pretty good splice site. So we would call this G the last of our exon. Remember, this is in frame 2. Plus 2. So, if that's the case, this means that this exon would end in phase 0 because it's the end, it's frame 2, so it's the end of the codon for this serine here, right here. So we're going to call this 16156. This is on the positive strand, so all of the exons in a particular gene will always be on the same strand. This one is in frame 2, and it ends in phase 0. And remember, there's always a comments column here, so if you want to make any notes about anything to do with the annotation, you can go ahead and do that. And also, just a reminder, in this workbook, I'm doing this under the Unique Exon tab. So we did this exon 2, exon 4. Remember, make sure that you're putting the coordinates in the right row, because then if we look at our RB, it'll tell us that we've done all of the exons so far. So there's 3 exons left. in isoform B. What I'd like you to do is go ahead, and this is going to be this one, and this one. Go ahead right now. You can pause the video. Go ahead right now and see if you can annotate those last 3 exons. So pause it. And you're back. So I'm going to put in the results that I came up with. Now, I'm not actually going to fill them in the worksheet here, but you've come up with, I'll just put in the start and the stop sites. So you might have come up with something that looks like this. And 16972, and then the last one, maybe you came up with 17027 to 19516. So let's say you came up with all your coordinates. These are all going to be on the plus strand, of course. So let's say you came up with all your coordinates for isoform B. Well, now what we need to do is we need to check them. To do this, we use Gene Model Checker, which is that 4th website. And this is the URL up here. An important note about Gene Model Checker. This does not tell you that what you determined was correct. What this tells you is that it's possible. So what Gene Model Checker is going to do, it's going to check the sequence and make sure that the coordinates that you chose can potentially work. So that it's going to check to make sure that there are no stop codons in the middle of your exons. It's going to check to make sure that you're using the correct nucleotides for your splice sites. So it's checking that you're following all of the rules of annotation. Again, this is still just a best guess. We don't know that your prediction is correct. We just know that you have data to support this as the best prediction. So how do we use Gene Model Checker? So the first thing we have to do is we have to upload our sequence. This is our unknown nucleotide sequence. So we're going to click on Browse. And this is the same one that you uploaded into BLAST before, because this is the sequence that it's going to check against. Under Errors and Consensus Sequence, you're going to say no. Once in a while, we do get contigs that have sequencing errors. If this happens, we'll deal with that on a case-to-case basis. This is very rare. Ortholog and Drosophila melanogaster. This is the name of the gene that you're annotating. We're doing CI, and we're doing isoform B. And this is important to put the right one in because this is what it's going to compare it to. It's going to compare it to the Drosophila version, the Drosophila melanogaster version. Here, you're going to put in the coordinates. So we're going to type them in just like this. So one number dash the other number for the first exon, comma, space, 15519 to 16156, comma, 16231 to 16743, comma, 16808 to 16972, comma, 17027 to 19516. So all of the exons for the isoform you're going to put in here. Annotated untranslated regions, this is going to be no, we're not annotating untranslated regions. Orientation of gene relative to query sequence. If this was all on the plus strand, as this was, then we're going to put plus. Completeness of gene model translation. This is going to be if you annotated all of the exons, then you put complete. You'll notice that the coding exon coordinates, the last exon, did not include the stop codon. So the stop codon actually went to 19519. So it was 19517 to 19519 was the stop codon. We don't include that in the coding exon coordinates. That goes in the stop codon coordinates. Now, when you click on it, it's going to automatically include the last 3 nucleotides as your stop codon. So this is important. Don't include the stop codon in the coordinates of your last exon. We put them in here. And then the last things we have to put in are project group. So that is what we are doing here. And we're doing the Drosophila erecta dot chromosome. So we're going to click on that. And project name, this is the name of the contig. So we're doing contig 38. We just type it lowercase, no space, just like that. And then we click on verify gene model. And if everything's correct, or if everything is plausible, you're going to get all green check marks and pass right here. And I'll talk about what these are in just a second. This is where it's important that you keep good notes and you're very precise in the nucleotides that you're choosing. If you are off by 1 nucleotide, so let's say instead of 156, you say 155 here, you click verify gene model, it's going to get mad at you and you're going to get these fails. So here it's telling us, and we know that we put the wrong number in here, it's telling us that the donor or the 3' splice site is not correct here because it's not an AG. It's saying you're using a GG for a splice site, and that's probably not right. It's also telling us that because you put in the wrong sequence, we're getting premature stop codons in our next exon and in exon 5. Because if we change the exon by 1 number, now it's out of phase for the next exon. And now everything's going to be wrong from there on out. So this is the same as having frame shift mutation. So it's important that we put our numbers in correctly. Now, if you do get an error, something like this, where it says fail, you can always click on the magnifying glass and it'll show you where in your genome browser that issue is. And you can actually use this just like, you can click on this and look at some of the different things. So I'm going to put this back to the correct number here. And you'll see it's going to check for the correct start codon. It's going to check all the 5' and 3' splice sites. It's going to check to see that the number of exons is the same in your unknown species as Drosophila melanogaster. Sometimes they differ. The vast majority of the time they're going to be the same. So if we want to look at the model for how our gene was annotated, you can click on any of these magnifying glasses, and it's going to bring up the genome browser, and you can kind of zoom out here. And up here in red, it's going to say custom gene model. This is going to be a visual representation of what you annotated for your isoform for this gene, right there. So it's a good thing. It's always good to kind of take a peek at it, make sure it looks legit. There are a few other things that this is going to give you that you're going to need when you submit your annotation report, when we upload everything to the main consortium. The first of these is a dot plot. So you can click right here. It says dot plot. What a dot plot is showing is this is an amino acid alignment between Drosophila melanogaster, which is on the x-axis here, so the CI gene isoform B, and the amino acid sequence of your predicted, of your unknown sequence, of known genome. So here you see there's these different color boxes. The boxes represent different exons, so there are 5 exons here. So anytime an amino acid is the same in both of these, kind of go, so like say amino acid 600, 600 here, 600 here, if it's the same, you're going to see a dot. If they're different, like say amino acid 700 here versus 700 here, there will not be a dot. So when you see dots, and a bunch of dots in a row, it means that's levels of high conservation. So exon 1 was really short. You can see here. Exon 2, there's a lot of conservation there. Exon 3 and exon 4, a lot of conservation. Exon 5, there are a few areas of differences. So if we were to look at this area right here, we would probably find that the amino acid sequences are somewhat different. This is fine. This is normal. What you don't want to see are very big shifts. That likely means that you missed part of a sequence. If you click up here on View Protein Alignment, this will show you the same thing, but in a protein alignment view. So the top row is going to be the Drosophila sequence. The bottom row is going to be your amino acid sequence for your particular species. Every time the colors change, this is a new exon. So this is exon 1. This is exon 2. Then exon 3. So every time you see a color change, so red versus blue here is exon 2 of Drosophila versus exon 2 of your species, and then orange versus light blue is exon 3 versus exon 3, and so on. And you can, if you remember from the dot plot, I'm going to close that. There were some regions of non-homology here, so somewhere around 700 or so. So if you look at the protein alignment and go around 700, you can actually see that here. There are these regions where there's not a whole lot of amino acids that are identical. And this can be informative about the function of the protein. Regions of proteins that tend to be highly conserved among different species oftentimes have functional regions that are important. So if you were to look at this region here where there's not a lot of conservation, it might be unlikely to find parts of this protein that are involved in some sort of function like DNA binding or phosphorylation or some sort of activity. But regions of this protein that are very highly conserved, so like here, for example, we might find, and especially down here, we might find that these are functional parts of the proteins. And later on in the semester, we'll talk about ways in which we can look for those. So this is our protein alignment. And we will need to save these files because they do go in our final report, and we'll talk about how to do those as we get closer. So transcript sequence, this is going to tell us what the the sequence of transcript is, the peptide sequence, and then under downloads, these are files that have to be uploaded with our final submission. I'm not going to talk about how to put together the final submission document because we'll talk about that once we get a little closer. What's important now is that you're learning how to put together your annotation. When you get to the end of your first isoform, let me know. We'll talk about the best way to save these files so that when you do have to put together your annotation report, you can do it relatively quickly. So that is how we annotate these genes. This is a relatively simple gene. There aren't too many surprises with this one. You may find that your genes are a little more complicated. That's fine. You have plenty of time to work on these. and I'll certainly be around to help. If you've had any issues with this practice gene, let me know. We can work on it. I want to make sure that you have a good grasp on how to do this. And if you want, you can always practice one of the other isoforms of this particular gene. You can practice any gene you want because you can check any gene you want in Gene Model Checker, especially anything from this contig because you've already got that sequence. So you've got enough information now to start annotating your own contig. You've only annotated a few exons, but you use the same process for all of them. One thing I am going to say, since I don't have a chance to hand these out because we're not in lab, but if you go to the Google's or the eClass, under annotation resources, there are these different resources that are useful. So the first one that you might want to look at is the annotation instruction sheet. This is a longer document. It's about 9 pages. It gives you kind of the ins and outs of annotation and some basic guidelines. I'm not going to go over this specifically, but I'd highly recommend you read it because these are some things that you may run into when it comes to things like searching for short exons and what's the important conservation of gene structure. So I'd recommend going through this, and if you have questions about it, let me know. So that's the annotation instruction sheet. The second is the GEP annotation flowchart. That's this one here. This is a good thing to keep around. This is the overall workflow for anytime you're going to annotate any particular gene. So remember, the first thing you do is you look at if there's a gene prediction by one of the prediction programs. You click on that prediction, you click predicted protein, and then you blast it in Flybase. You take a look at it in the gbrowse, and then you go to Gene Record Finder. You start mapping the exons, just like we did. This is your workflow. This is a good thing to keep around. The nice thing about this is each of these colors corresponds with which program you're going to use for those parts. So if you feel like you're not sure, you're lost, you're not sure where to go, take a look at this workflow. It's oftentimes very helpful. The last one for right now is determine CDS flowchart. This seems kind of complicated, but this is really what we've already done for annotating an exon on the 5' end. Do you get an acceptor that's and this is the 3' splice site? Do you get one that's AG? If no, then you probably are going to have to look somewhere else. If yes, then you're going to look for compatibility with phase for the previous splice site, and so on. So this is what we did when we were looking for the splice sites. It also talks about what is the evidence that you're going to try to use. So we talked about gene prediction tracks, the BLASTX tracks that are there to look for conservation, RNA sequencing data. Clustal alignment is something that we'll likely do later. So if you find this flowchart helpful, you're welcome to use it. So the next step is to start annotating your own contigs. So to do that, you're going to need to know what your contigs are and who your partners are. So I've put together 4 pairs of people. I've done the very best I can to match up people with who they wanted to be with. So here are the groups right here. Let me zoom in a little bit. So Heather and Katie, Jess and Ken, Pat, Nicole, and Alexa and Kelly. each group is going to start with a contig. So this is the contig that each group is going to start with. So Heather and Katie are going to start with 33, Jess and Ken with 6, and so on. If you go to eClass under contigs, there are 4 folders. So if you're working on contig 6, you can open that folder. There are going to be 2 files. One is going to be the FASTA file. So this is the sequence. You're going to need to download that, and you're going to use this for your BLAST and for your gene model checker. The second one is the project report file. You don't need this yet. We'll use this later. So all you really need is this FASTA file right now. So let's say you've got contig 6. So let's say you're Jess and Ken. You've got contig 6. So where do we start? Well, you're going to go to the genome browser page, and the species we're using is Drosophila ficus phyla, so right here. And the assembly we're using is this GEP3L control. So what this means is we're actually annotating part of chromosome 3 on Drosophila ficus phyla, because for each of these new genomes, we need to annotate not just the ring chromosome, but the other parts of the chromosomes as well. So we're going to be annotating the L just means left, so it's the left arm of chromosome 3 from Drosophila ficus phyla. So we're going to type in contig 6 here, and it's going to pull it up. You're going to see it's a little more complicated than. I'm going to turn off these splice acceptors. I really don't like how it's keeping them full like that. So it's going to look a little more complicated than the one that you just did, but that's okay. You've got plenty of time. You can keep plugging away at it. You're probably wondering, where do I need to start? So for each contig, I've put a suggestion for a gene to start with. So for example, here, we might want to start with CG11137. So if we go here, you can see this is CG11137. This is where there's a BLAST-X alignment to it. So if you were going to start with this region right here, this smaller gene CG11137, if we go to our GEP workflow, what's the first step? Well, we need to select which predicted protein that is. So we would go, and this is just like we did with the CI gene. Maybe we choose this one here, this GenScan prediction, because it looks kind of like the BLAST-X alignment. So we're going to click on that. Here you actually have to click it twice, because clicking it once, it's just going to give you the information on contig. And then we're going to go to predicted protein, and we're going to copy this, and then we're going to blast it in the fly-based BLAST-P. And we're going to determine, is it a good match with the Drosophila melanogaster gene? If it is, then we can go to Gene Record Finder and start annotating those exons. So if you're not sure what to do, you can always look at the annotation flowchart. You can also always look at this document that we've been using. Look at what we did when we started annotating CI. So where we started on page 16 here, sample annotation, you're going to do the exact same thing. So all of these files are up on eClass. You have access to them. I'd like you to start plugging away at it so that when you come to lab next week, you've started to at least attempted to annotate the first gene in your contig. If you have any questions, if you're running into any stumbling blocks, please let me know. On Thursday, I will be around. You can always email me. And then you can always stop by anytime Friday or next week if you have any issues, because I'd like you to start getting going on this before we meet again next week. Good luck. Have fun in the snow tomorrow.